Hello all, this is chapter 26 of Biotechnology and Genomics. So biotechnology is use of natural biological systems to create a product or achieve some other end. DNA knowledge enabled of gene manipulation, and scientists can modify genomes through genetic engineering to improve an organism's characteristics, make biotechnology products, or treat cancer and genetic disorders. So there are cloned genes that are used to alter the genome of viruses or cells. A GMO is a genetically modified organism that has a modified genome. An example of this is a transgenic organism and had a gene from one species from another species foreign to it inserted into its genome. So cloning is reduction of identical copies of an organism, cell, or DNA through asexual means. So examples would be bacteria from the same colony are identical twins. And gene cloning is reduction of many identical copies of a single gene. So one of the uses of gene cloning is to produce large quantities of insulin, to make GMO organisms, and to perform gene therapy to treat human disease. So recombinant DNA or rDNA contains DNA from two or more different sources. To make rDNA, we need a vector, a piece of DNA that foreign DNA can be added to. Plasmids are accessory rings of DNA in bacteria commonly used as vectors, and they are not part of the bacterial chromosome. So here an example of cloning a human gene. So we have human DNA inserted into a plasmid. That plasmid is then inserted into E. coli. And so we have many different copies of that gene, and it can be used to make a protein such as human insulin in bacteria. So there are two enzymes needed to introduce foreign DNA. The restriction enzyme is to cut open the plasmid, and then we need DNA ligase that will seal the foreign DNA into the opening created by the restriction enzyme. After the cutting, a gap is created in which pieces of foreign DNA can be placed if there is complementary pairing. Foreign DNA and the plasmid DNA or vector DNA are cleaved with the same restriction enzyme, and single-stranded complementary ends of the two DNA molecules are called sticky ends because they can bind to each other by complementary base pairing. And sticky ends facilitate the insertion of foreign DNA into the plasmid or vector DNA. And DNA ligase is an enzyme that functions in DNA replication as used to seal the foreign piece of DNA into the vector. Here's a diagram showing how a restriction enzyme cuts open a duplex or double-stranded piece of DNA and has overhangs that are known as sticky ends. DNA sequencing is a procedure that determines the order of nucleotides in a segment of DNA. It helps identify specific alleles and sequences, and we can facilitate development of disease treatments and can reveal evolutionary history and relationships. And it's a foundation of forensic biology. And dyes are attached to nucleotides, can show order of nucleotides in automated DNA sequencers. And many copies of the DNA segment or gene to be sequenced have been made using the polymerase chain reaction. So the polymerase chain reaction, or commonly called PCR, can create billions of copies of a segment of DNA in a test tube in hours. And it amplifies only specifically targeted DNA sequence. And the targeted DNA sequence is usually a few hundred base pairs in length, though it can be up to a few thousand, and uses DNA polymerase and DNA nucleotides to G's, A's, T's, and C's. And three basic steps that occur repeatedly, usually about for 35 to 45 cycles, and this usually takes about 3 hours. Sorry, I skipped a slide. So this is an example of the PCR chain reaction. And it occurs in a thermocycler, and you have the denaturation where double-stranded DNA is converted to single strand by heat. Primers anneal, and that's the annealing step. And the third one is extension, and that's where Tac DNA polymerase makes its copies. PCR is a chain reaction because the target DNA is repeatedly replicated, and the amount of DNA doubles with each cycle. And automation is possible because the use of a temperature-insensitive DNA polymerase extracted from Thermus aquaticus, a bacteria that lives in hot springs. And this enzyme tolerates the high temperatures used to separate the DNA strands. 95 degrees Celsius are almost boiling. DNA analysis has improved over time. Previous method involved the entire genome being treated with restriction enzymes. Fragments were separated by gel electrophoresis, where the smaller fragments move faster than the larger ones, and its result was distinctive patterns of bands called a DNA fingerprint. So here is DNA fingerprinting by gel electrophoresis. So there's a PCR reaction on repetitive sequence in the DNA. So we have DNA from the crime scene, suspect A and suspect B. We can see suspect B matches up with the DNA at the crime scene, so that person was most likely there. So for DNA analysis, they use short tandem repeats or STRs. Like the name sounds, these are same short sequences of DNA bases that repeat several times. So that it's a tetramer there, gotta. And fragments in samples amplified by PCR are different lengths because we have different numbers of repeats through different people. And so the more STR loci employed, the more confident scientists can be of distinctive results for each person. For STR profiling, PCR is used in fluorescently tagged sequences, and the products are run through an automated DNA sequencer. A laser detects and records the length of these DNA fragments. The greater number of STRs at a locus, the longer the DNA fragment will be, and this can be used to identify samples at a crime scene, detect genetic disorders, identify relatives or victims of a crime. Genome editing is a relatively new advance in DNA technology, and it targets specific sequences in DNA for removal or replacement. There are several methods, but the most popular one used is CRISPR, or clustered regularly interspaced short palindromic repeats. So CRISPR was first discovered in prokaryotes, acting as immune defenses against invading viruses. And the system involves an endonuclease enzyme called Cas9 and identifies specific nucleotide sequences in genomic DNA of the invading viruses, breaks both strands, and inactivates the virus. It also uses a guide RNA molecule that's complementary to these base pairs to the genomic DNA sequence. So Cas9, scientists can study a gene's role in the cell after Cas9 break and activates the gene, and it can also insert new nucleotides at specific DNA locations. So CRISPR can target specific sequence of nucleotides for editing in almost any organism. You don't need to know the details of this, but this is just a diagram of how CRISPR and the RNA guide molecule works together. So CRISPR applications include treatment for diseases such as sickle cell disease and cancer, and is also used for rapid tests for the COVID virus. And scientists are continually working to make the system more efficient and develop new applications. So here's a diagram showing CRISPR using to fight sickle cells, to fight cancer, for editing sperm, and is involved in vitro fertilization therapy. So this is a section on biotechnology products. So it's about transgenic organisms, such as GMO crops. So transgenic bacteria are produced by recombinant DNA technology and grown in large vats called bioreactors. They express the cloned gene, so we can make insulin, human growth hormone, TPA, Hep B vaccines using these techniques. So other uses of transgenic bacteria, bacteria live on plants have been altered for frost plus to frost minus bacteria. So we have frost resistant strawberries. There also makes it, we have bacteria that may be toxic to insects that damage the root of corn. And here we have transgenic bacteria that can help eat up oil and sulfur from coal and oil spills. For GMO plants, foreign genes can be introduced into plant embryos called protoplasts. And so they are exposed to an electric current when the liquid containing the foreign DNA, the pores seal up, and then we have mature plants that express the foreign gene. So we have potatoes below ground. We have potatoes that below ground and tomatoes that are above ground. Pest resistance in cotton, corn, and potato strains, soybeans to herbicide. And we have plants that can be engineered to produce human proteins. So here's a GMO of apple on the bottom. It doesn't turn brown as quickly as the normal non-GMO apple on top. We have GMO animals. So this is technology where we microinject eggs and then when eggs are fertilized, we have transgenic offspring. So BPH or BGH, bovine growth hormone, has been inserted to produce larger fish, cows, pigs, rabbits, and sheep. We have the Aqua Advantage salmon. All right, so they grow. So these reach adult size 3 times faster than wild type salmon with less food. See, here's an example of these transgenic salmon compared to each other. So transgenic farm animals can be used to produce pharmaceuticals, and the protein can be harvested from the animal's milk. And this is how a diagram of how you produce transgenic animals. You microinject the DNA into an egg, a donor egg, place it in a goat, and then you milk the goat to get the protein. So we've also cloned animals before. So remember in 1997, Scottish scientists cloned a sheep named Dolly. Since then, calves, goats, pigs, rabbits, cats, and dogs have been cloned. The process is difficult, but it can be done. So this is the cloning process. Donor eggs are microinjected with nuclei from one transgenic animal, then to begin development in vitro. Development continues in host females until the clones are born, and the female clones has the same product in their milk as the original transgenic animals. So here are transgenic animals of making human growth hormone in the milk. So through biotechnology, we also use gene therapy. So we're trying to cure genetic disorders. So we use viruses and liposomes to put foreign DNA into cells. Sometimes the gene is injected directly into a specific region of the body. All right, so these are different ways, or different gene therapy techniques and ways to get them. So in lungs, they use an aerosol spray, for example. Some people inject it right in the bone for the bone marrow. All right, so if you have muscles, you can inject it directly to the muscle cells. So you can do ex vivo gene therapy where you take cells out of the body, you change them, then you put them back in the body, and this is how they treated SCID patients. And this is a diagram of the SCID patient's treatment. We remove the bone marrow stem cells, you treat it with a retrovirus, and you put it back in the bone marrow. So here's another example where they take a piece of the liver out, place a virus in it with the normal gene, then that liver is returned to the patient. So we have in vivo gene therapy. So for example, for cystic fibrosis of, the virus containing the transgene or the new gene is sprayed into the nose or in the upper respiratory tract. This is also how we treat cancer using, find a way to introduce tumor cell gene suppressors, such as P53 into cancer cells, or make the tumor cells more sensitive to chemotherapy or X-ray radiation. We can use what's called RNAi or RNA interference to silence expression of specific alleles, where an RNA molecule targets another message to knock down that mRNA so that protein is no longer expressed. This was first discovered in worms, and there's lots of research on this currently ongoing. So genetics in the 24th century is known as genomics because we can sequence the whole genome of humans in a day. And then we mapped all the genes as well, 20,000 genes that code for proteins in humans. Before next generation sequencing, there was a 13-year effort to sequence the whole genome. It took 13 years. But now, because of new technology, we can actually sequence of your whole genome in a day for about $5,000 if you want it. So because of the sequencing of the genome, we discovered these single nucleotide polymorphisms, also known as SNPs. And they may have no effects. Sometimes they do have effects. And this allows to raise the possibility of designer drugs tailored to an individual's genotype. So when we sequence the human genome, there are structural genomics, knowing the sequence of the bases and how many genes we have. And there's also functional genomics, like what does it actually code for? And we also found out that non-coding, or what they used to call junk DNA, may have important functions that we just realized. So the genomic architecture, 98% of our human genome is DNA that does not code for proteins. Some is transcribed into ribosomal RNA or transfer RNA, which are both involved in protein assembly. And the rest of the genome consists of a variety of sequences. Some are repeated and others are not. So we also found there's lots of transposable elements or transposons that jump around in our genome, and this is 44% of our human genome, and it's thought to drive evolution. And we also have lots of repetitive DNA elements, the same sequence of 2 or more nucleotides repeated. And so it may not be useless. It used to thought to be non-important, but it might control telomeric function. And of course, we have sequences with unknown function. So what is a gene? Historically, a gene was thought of as a particular location or locus on a chromosome, and eukaryotic genes appear to be randomly distributed along chromosomes, and they're fragmented into exons and intervening introns. And 95% or more of most human genes are introns, and they may be the regulators of gene expressions. And exons, which is a coding sequence, may be put together in a variety of ways to make different protein forms. So a more modern definition focuses on transcription, and a gene is a genomic sequence that directly encodes functional products, either RNA or protein. And a gene may not be found in a particular locus on a chromosome, and genetic material may not only be DNA. Some prokaryotes have RNA genes. So we have the area of comparative genomics, where we compare genomes of different organisms and looking for similarities. And we can see the way genome changes through time. So for example, tracking the evolution of HIV virus, and we can understand the evolutionary relationships among organisms. So, for example, human and chimpanzee DNA are 98% identical or alike, and human and mouse are 85% identical or alike. So here is a table showing, different organisms and the size of their genomes, the number of genes they have, and the number of chromosomes. So functional genomics is to understand of how these genes interact, and we can use DNA microarrays to tell which genes are turned on in a specific cell or tissue and which are not. And we can get a person's genetic profile using DNA microarrays to identify various mutations to determine if certain genetic diseases are likely. There's also the field of proteomics, which is a study of the structure, function, and interactions of cellular proteins. And proteins differ depending on each cell type, and each cell produces hundreds of different proteins that can vary between or within cells depending on the conditions. And we can also use computer modeling to predict a three-dimensional shape of proteins, so we can figure out what drugs will target them. So another emerging field is bioinformatics, and it's the use of computer technologies of specifically developed software, statistical techniques to study biological information and create databases for the genomic and proteomic of experiments and results. And so bioinformatics can find significant patterns in the raw data of DNA sequences, and computer programs can help make correlations between genomic differences among large numbers of people and disease phenotype. The last part and section of this chapter is the BLAST tool in bioinformatics. So it stands for Basic Local Alignment Search Tool, and it's used to line up two DNA sequences or protein sequences to look for regions of identity or similarity between different organisms. And that way we can figure out what is the same and what is different. That is the end of this chapter.